Right. So we are going to start now. So today's topic is molecular biology techniques. Sorry for the date. I'm Dr. Megha. Right. So initially I was about to take the part two, but then I realized that I think it would be more prudent that we first cover the part one, which is actually the basics of it all and which deals with all the principles and fundamentals. So, you've been in microbiology and you've been talking about laboratory diagnosis. And so far, what you all have done is talked about laboratory diagnosis in bacterial, viral, fungal, among basic principles. So, this is the basic format when you get a question, and that's how you write the answer. You first talk about direct microscopy or direct demonstration, right, where you talk about staining, and then you talk about culture, where you can grow that organism, the bacteria in a culture plate, or a virus in a special cell culture medium, or you talk about antigen detection by various methods which are available, as well as antibody detection in serum with the available assay, and most commonly used are ELISAs and immunofluorescence assay. Uh, in that line, the next point we always talk about is molecular assay for detection of DNA or RNA. Nowadays, while this is the traditional sort of sequence that we have always gone about in laboratory diagnosis, for many pathogen, molecular diagnosis is now the first assay that now we go to. It's now become the most sensitive and specific assay for many of the pathogens. So, dealing about molecular methods, first we talk about why do we need molecular techniques. Because the traditional methods all have certain lacunae which has been missing, and molecular techniques sort of fill that gap, right? Because many pathogens are, we know that they are non-culturable, they're very fastidious, that is, they require certain special ingredients for to grow. Some are very slow growing, right? If a TB culture can take 4 weeks, you cannot wait 4 weeks for your diagnosis and for therapy. Some agents are highly infectious, you actually cannot culture them, right? So they require special facility or BSL levels, biosafety levels, and these are specially designed labs where you can handle these organisms to grow them, right? You cannot grow them anywhere. But for detection method, where you are not amplifying the organism in culture, remember, the organism gets amplified. That is why it's not safe to handle in an open environment. So these infectious agent, maybe molecular assay can be done, which are lower in the level of infectiousness, right? And also there is a problem that in many of the samples, the pathogen is available in a very low number. So for example, a sample like CSF in a case of meningitis or encephalitis actually have very low number of pathogens. By using these traditional methods, we actually are not able to detect them. And sometimes even the sample which is available, or specimen which is available, is very minute quantity, and you cannot actually use all the methods, right? Same example, CSF, for example, when you're doing a lumbar puncture, the volume that you're getting sometimes is actually very small, in probably in microliter or in ml, right? Also, there is a problem that sometimes there are agents which belong to the same family or same genera. They are very antigenically similar. So using an antigen or an antibody based assay which uses those antigen actually becomes problematic because you cannot differentiate them, right? Molecular assay is also important for molecular epidemiology. Now it is the foremost means of doing classification and nomenclature of organism, right? And also sometimes when you grow something which is new and which cannot be identified by traditional method, you actually require you need to confirm them using these molecular methods. Just a brief overview about the DNA structure. You have already more familiar with it, and you've already covered it in your last year in biochemistry. So DNA is deoxyribonucleic acid, which has these 4 nitrogenous bases, purines and pyrimidines, A, T, C, G, as we talk about. Nucleoside is the nitrogenous base plus the sugar moiety, right? And nucleotide is when a phosphate structure is also attached to it, which gives it a negatively charged we know that DNA structure is a double helix structure. The 2 strands are antiparallel and they are complementary to each other. One runs in 5' to 3', other completely opposite to it. The A, T, C, G are bound to each other. The A and T forms a double bond and C and G forms a triple bond. In DNA replication, you guys are already aware that there are three enzymes which are commonly used. You have DNA helicase, which first separates the two strands, and these two strands, one is called the leading and other is called the lagging strand. DNA polymerase is the enzyme which actually initiate this strand placement, and DNA ligase is the one which actually binds the interrupted sequences of DNA in the lagging strands, which are called the Okazaki fragments. Why we have just gone over the DNA basics and DNA replication? Because all this knowledge actually has been used to create the newer molecular techniques that we are going to talk about in the next few minutes, right? This is the basic central dogma that you already know about. DNA, which forms the RNA through transcription, and which forms the protein, which is the translation, right? So these already familiar knowledge has been utilized further. Before moving on to the principles and what are the methods of molecular techniques, let's talk about the first step of getting that template DNA, right? So when you have a sample, it has everything inside, not just the DNA and RNA. It has the cell itself, it has proteins, it has carbohydrate, it has lipids, all kinds of molecules are in there which can interfere with your molecular technique that you're going to use, right? So first step is actually from the sample to extract the DNA and RNA separate, right? So this method is called nucleic acid extraction or isolation. So in this method, we release the intracellular DNA and RNA, it reduces the interfering substances, and it makes the analytical method more reproducible. The different samples which have been used, actually the extraction method will also differ slightly because there are some samples which have more interfering substances, like if you take a stool sample from a patient, right? Or you take a sample which is a sterile fluid. They all have different formulations of different requirement for a extraction method, right? So different samples could be blood samples, serum, plasma, any of the body fluids, right? Which could be mostly sterile fluid or a non-sterile fluid as well, right? Like urine. It could be tissues, it could be cytopathology sample, it can also be culture colonies or the cell culture lysates, right? So a extraction method involves three steps. First is the lysis step, then there is denaturation of proteins, and then there is separation or concentration of DNA. In the lysis, you actually lyse the outermost cell wall, membrane, envelope, any coating which is there using different ionic detergents, alkalis or enzymes. Next, you'll need to denature the protein. So we have different, it can be done through chemical methods or through enzymatic method. Guanidinium isothiocyanate is one of the common substance which has been there in many of the extraction kits which denatures proteins. Then separation and concentration of DNA can be done by various methods, precipitation methods, liquid phase extraction or solid phase extraction. When first extraction method started, precipitation method was the one which was first devised. In this, what we do is first we concentrate the protein or the precipitate out the protein using high salt concentration liquids, and then we precipitate the nucleic acid by adding ethanol or isopropanol. Then we sort of devised newer method where we use liquid phase extractions, where a phenol chloroform isoamyl alcohol compounds are used for extraction, right? These various substances are able to disrupt membrane, denature protein, and are able to concentrate or separate out the nucleic acid, right? So these steps, when they had come, they were mostly in-house methods, right? That means that the laboratory had to procure these reagents separately and devise their own methodology, standardize their own protocol, and perform these extractions. Now what has happened is most of these are available in a kit formulation, right? And the third step, solid phase extraction, is the one which is now commonly used, right? It's easy to do and it doesn't require this standardization or procurement of different reagents by the lab itself, right? In solid phase, as the name suggests, there is going to be a solid matrix, which is the immobilized component where the nucleic acid will bind, right? And then this nucleic acid can be separated into another tube or a vial and can be used for further downstream application. This solid phase can be a silica membrane. There are different other compounds which are also available, like hydroxyapatite, iron oxide. This is just for enlisting, but most commonly used are actually these silica membranes, which has been tried. Just giving you an example of this. In this, you actually have this tube, and in this tube, the lowermost part has a small silica membrane in it, which is covering it, right? This is a small cylindrical tube with one end closed and other end open, right? So in this, what we do is, when we add the sample, we first add all the lysis reagents, which will remove all the cell membranes, the coats and everything. And this silica is negatively charged. So it will generally, as the DNA or RNA, they are actually negatively charged. So in a normal neutral solution, it will repel DNA, right? But when you change your buffers, you actually increase your sodium chloride concentration, you add different solvents, this nucleic acid is forced to bind to the silica membrane while making the sodium salt bridges. We also add ethanol in it, which actually improves this binding. And subsequently, everything else, so now the DNA or your nucleic acid is now bound to your silica membrane, and above it is all the non-bound particle. Only the nucleic acid has bound, so all the carbohydrate, lipid and other protein materials are unbound. So then it goes through several washing cycles where all the unbound particles are removed, and after that you change the buffer. Now you add a low salt buffer in it. So now what will happen, as was happening earlier, the silica will now repel the DNA, and this DNA now will come out into the newly added buffer. This buffer is then, through a centrifugation process, is collected at the bottom and is then separated and stored for further downstream application. So just to tell you that in a little more clarity. So first you have the tube where you've added everything. The lysis step is happening. The nucleic acid is separated. The protein is being denatured. The membranes and other things are being lysed. When you've added it to this tube, which has the silica membrane, there is the nucleic acid and all the unbound particle inside it. The nucleic acid will bind to the silica membrane because you've added the high salt concentration containing buffer. And then it will go through several wash cycles where all this unbound material is going to be washed away, right? So at this stage, only the membrane with the bound DNA is there and everything else has been removed. Now you'll add another buffer, which is a low salt containing buffer, and here now this DNA molecule will come into the buffer, and when we'll centrifuge this, it will come down and settle at the bottom, and now we'll store this part, right? So this is how DNA extraction using a silica based membrane is being in use, and it is actually conveniently available in a kit formulation, which nowadays most of the labs are using. Now one is doing all these steps separately by a lab, and this whole process has now also been automated. So you have these machines where you can load all your reagents, put the tubes inside. There is a robotic arm which will do all that pipetting, and at the end you will just get the eluted DNA and RNA in a tube, right? The advantage of this is these are high throughput machine. That is, at one go you can actually get many samples together. When you're manually handling, you can only do a limited number of samples, but using these automated machines you can actually do large number of samples together, right? So you can actually test greater number of samples. These automated machines use similar principle like the silica membrane based principle that we just talked about, where the separation part is actually being taken care of, where earlier we were centrifuging. Here we are using a vacuum to pull down all content at the bottom, right? Other than the silica based membrane, many of the automated systems available also have a magnetic bead based system as well, right? These two machines I've just shown you, these are from different manufacturers. They have these magnetic bead based system. So to just tell you a brief, the similar steps that we talked about has been happening here as well. the sample which has been completely lysed, but there where a silica membrane was there to bind to the DNA, here what they have done is they have added some magnetic beads over which the DNA molecule are actually being adhered due to the buffer concentration. And we add a magnetic probe here. So what will happen is all the magnetic beads, when you will charge it, all the magnetic beads will get attached to that probe, near to that probe, right? And all the unbound material, which is the non-DNA component, will be in the liquid separated, right? And then this component, this with the probe attached, will go through several wash cycles. All the unbound particle would be removed, and finally you will remove the magnetic charge, and the magnetic beads will then come out and it will separate, and all the DNA nucleic acid will come into the elute, right? This is actually very interesting because you can actually see those black colored magnetic beads sticking to the magnetic probe, or being concentrated at the one end, right? And all the washing step being continuing, right? So again, there's similar advantages to that. So these are the common methods by which we are purifying the DNA, and we are then using it for various molecular techniques. Now coming on to those molecular techniques, there are various techniques which have been used. You have nucleic acid hybridization assays, you have nucleic acid amplification methods. These amplification methods are the one which are now most commonly used. There are two types. One is target amplification and signal amplification assay. Target amplification is the one where the target gene is being amplified and then detected, right? PCR or polymerase chain reaction is the most common technique which is being used, and it is a target amplification based molecular technique. So in PCR you have alternate cycles, that is alternate cycles of temperature being used. So there is one modification where this whole target amplification is done at a single temperature. So these are called isothermal amplification assay, which are also available. So NASBA, TMA and LAB, there are various techniques. It's okay to know the names even if you don't know the details of each and every technique. So you have nucleic acid sequence based assay, TMA is transcription mediated amplification, LAMP is loop mediated isothermal amplification assays. So these are different assays which are available. Every technique has its own advantages and disadvantages, right? We cannot go in detail about each and every one, but we'll go over the most important ones, right? The other techniques are signal amplification assay, where the known ones are LCR or ligase chain reaction, and bDNA or branch DNA assay. Among all these, again, PCR is the one which is being most popular and also easily available and easy to perform. Then there are nucleic acid sequencing, which can be performed. Sequencing is identify the exact nucleotide code of your DNA. There are different sequencing methods which are there, like a dye-terminator method, pyrosequencing, and now we talk about NGS or next generation sequencing, identifying whole genome sequence, WGS, right? So these are several newer techniques that has come up. Cloning is also one of the method which has been used as an aid to create controls while you are doing another assay, but it is mostly a research technique, right? Not for a diagnostic base. Now there are CRISPR based molecular diagnostic methods which are also available. If you must have heard in news, CRISPR is a newer technique which has come up, right? It is clustered regularly interspersed sequence palindromic repeats and short palindromic repeats, and you must have heard it in news because two of the scientists, women scientists who were actually devised it, recently also got Nobel Prize for it, right? So first let's talk about polymerase chain reaction. So PCR, it was first discovered by American biochemist Carey Mullis, and he devised it in 1986. So initially when it had come, it though immediately was not the most preferred technique. At that time the traditional methods were being used, but subsequently this has become a central technique in biochemistry, molecular biology, microbiology and infectious diseases. He also received a Nobel Prize in chemistry in 1993. So PCR, we talked about, it's a target amplification assay. It combines the same principles of complementary nucleic acid hybridization with nucleic acid replication, right? Applied through repeated numerous cycles. It's like a DNA photocopier which will copy a nucleic acid target and multiply it to several times, generally 10 to the power 7 or more in a very short period. It provides ample target which is then readily detected by various other methods. Now understanding this, again you I think already have a little introduction to PCR and biochemistry. So this is sort of again a more detailed overview of the same thing. So you have the original DNA. This is what you are trying to amplify. There are various milieu and other things which you have added into its mixture. These strands are first separated, the primers bind to it, and then these amplify. This follows through these 3 cycles or steps which are called denaturation, annealing and extension. Right? So this is a common question for you, PCR and its steps, when somebody talks about it. So denaturation is the first step where the sample is heated to more than 90, about 95 degrees Celsius. This denatures or separates the 2 strands of double-stranded DNA. Right? Next step is the annealing, where the temperature is lowered. Generally, it is brought down to below the melting temperature of the primers. Right? Primers are the short oligonucleotide sequences which are at the opposite ends of your target region. Right. So annealing is that it is bound to the primer has bound to your DNA. Next step is the extension, where your polymerase enzyme is working and it is actually making the new DNA strand. Right. So one thermal cycle theoretically doubles the amount of DNA which is present in the reaction, and it roughly take about 25 to 45 PCR cycles for a target to be amplified in appropriate amount. So what are the essential components here? You have your target sequence of interest, which we are calling template DNA. You have the primers, which are nothing but synthetic oligonucleotides, that short DNA sequences. There are 2 primers which are used. One is forward, other is reverse. You have thermostable DNA polymerase, which is catalyzing the DNA synthesis. It is one of the most crucial enzymes which has been used, which we'll talk in detail. Then you need the building blocks, which are the DNTPs and which are ATCG. And you have divalent cations like magnesium ions and others, because the enzymes require ions for its function. And you have the buffer solution. Right. So in a PCR, you have all these components which are added into the PCR tube. In the PCR tube, which first we call, when we add all these things, we call it master mix preparation. All these preparation with the template DNA is added into the PCR tube. This is a pipette, and it is pipetted into the PCR tube, and this tube is then loaded into the thermal cycler. Right? This thermal cycler will alternate the temperature, go through cycles of denaturation, annealing and extension, and amplify your DNA strand. Right. So just looking at PCR in a small video. So after you finish this lecture, if you have time, go to YouTube, watch a couple of videos. I think you will understand the basics of many of these techniques in very simple sort of small videos. So you can see where you have the sample, you have extracted the nucleic acid, the DNA, and you want a PCR to be done for this, right? So this is a thermal cycler which we are using, and it carries out the thermal cycling, that is changing the temperature frequently in a short period. This is your target gene in the DNA template. So you have the PCR tube where you have added all the ingredients inside the tube, and this tube is then placed into this thermal cycler, which has these small blocks where you can put the tubes in. So as PCR is happening, we can see that first, concentrating on the target DNA here, we can see as it goes through first cycle of denaturation, the temperature is high enough that the two strands separates, and this is all which is available in the master mix solution, which has been added, which has the polymerase, the forward and reverse primer, the nucleotides ATCG, and the ions and the buffer. As it goes to annealing temperature, the primers, which are the short oligonucleotide sequence complementary to opposite ends, binds to those regions, and then as it goes to extension, the polymerase will extend the strand using the same nucleotides. So this activity is being repeated several times in a PCR. In the next cycle, the steps are again seen in a similar manner, where denaturation, annealing, extension will further almost doubles the amount of DNA which was present previously. So one thing you will see here is not just the target sequence is being amplified, the area which is adjacent to it on one end is being extended. Why? Because DNA polymerase has a 5' to 3' action. So when it reaches the other end, it is actually being extended to the end of the DNA which is available to the DNA polymerase molecule, right? So you not just have the target DNA, you also have other DNA, but the length of the target DNA is much more at the end of each cycle compared to the variable length DNA fragment, right? So this is sort of an exponential growth that we are seeing here in PCR. So these cycles will take certain time as it's being set. As we earlier talked about, we generally go for 25 to 45 cycles. So from one single DNA, at the end of 4 hours, you actually get these many copies of your amplified DNA, right? Of your target DNA. A huge amount of target DNA and a low amount of variable length DNA. So at the end, we are now removing the PCR tube, and this is ready for your next part. Now what is the next part is the post-PCR analysis that we'll talk about just a few minutes later. Before that, among the components that we talked about, one of the most crucial component we said was tag DNA polymerase. So what is it? It is a 94 kilodalton protein and its source is Thermus aquaticus. So when they found these small lakes which were actually always at a high temperature, they found these special bacteria which are able to grow, right? And the DNA polymerase that they derived from these bacteria, Thermus aquaticus, was able to withstand a very high temperature. This molecule was actually the molecule of the year due to its enormous potential and importance later on. This has a 5' to 3' DNA dependent DNA polymerase activity. It has a set extension rate of 50 to 60 nucleotide per second, that is 3 kilobase pair per minute can be amplified here, and it can work at 95 degrees Celsius for about 40 minutes easily, right, to the period of the standard PCR conditions. Why is it important? Is because when first PCR was devised, initially the polymerase available, which were the one which were easily available in lab, which did not had this property of withstanding high temperature. So with each cycle they had to add a new polymerase, because every time it reached to 95 degree, all the enzymes used to be denatured, because enzymes are nothing but proteins, so they would be denatured, and at each cycle somebody needs to add the DNA polymerase, right? It was a very tedious process. So even in our lab also, because again we have not seen this being done, but our seniors actually when started doing PCR, that's how they used to do it. So at that time there was no thermal cyclers which can do this rapid change of temperature. So what they used to do is they used to keep 3 different water baths at different temperature, 95, 55, 72, and they used to be ready with the enzyme solution at one end. They used to add everything, the PCR tube, into one water bath, then second water bath, then third water bath. Every time it used to pass through 95, somebody had to manually open the tube, add a fresh batch of enzyme or polymerase, and then close the tube and put it back in the water bath, right? And remember, this is happening for about 40 cycles. So somebody is just sitting there and just putting it into different water baths and adding it, right? It used to be a very tedious process. It was not feasible to be used for diagnosis at that time. But presence of this or discovery of this enzyme actually sort of revolutionized the diagnostic application of PCR. So another important thing is thermal cycler. You can see these are different. This block has these certain wells where this tube can be fitted, or plates which has 96 well or 48 wells plates can be fitted. So this is based on Peltier mechanism. Read about it after this lecture if you get time. Right. So what happens after PCR? You saw that tube which came out of the thermal cycler. It had a clear liquid. So that clear liquid was when you put in, and after you take it out, DNA cannot be seen by naked eye. So you actually, even if it's amplified or not amplified, you cannot visually say that whether amplification has occurred or not, right? So post PCR analysis needs to be done after the PCR has been performed. The most common technique which is being used is agarose gel electrophoresis. There are other methods which are being used. Sequencing, RFLP, ELISA can be done, hybridization can be done. We are not going to go in detail because these are not the preferred technique. If we talk about specificity, right, which is like the most exact, it is actually sequencing and hybridization. So its results are the most specific, most ideal. But technically, these things are more difficult to perform and cannot be done in a routine diagnostic setup. So where agarose gel electrophoresis needs to be performed. Among the different PCR modification, one of which we are going to talk about is real-time PCR, which is now the most popular technique. This does not require a post-PCR analysis because here the detection occurs as the amplification is occurring. We'll talk about it in detail later. First, we'll talk about agarose gel electrophoresis. This is also something I assume that you guys have seen. If you have that visualization, I think it will be much clearer. So here a gel is formed, which is the agarose gel. Agarose is nothing but purified form of agar. So agar you must already be aware of. Agar is the substance that we use to solidify culture media. So most of your bacterial culture actually has agar in it. So this is something similar. It's been chosen because first it's been purified, so it doesn't have impurity and interfering substances. And the melting temperature is such that it's roughly about 65 degrees. So you can melt it. It can be brought to a liquid state. And then it rapidly cools. So at room temperature, it solidifies. So you can actually give these jelly-like structure can be made and set in a rectangular blocks, right? So this is what gel is. These are small combs which will create wells into the gel, and in these wells you can add your DNA, right? This DNA is then run through in electricity. We know DNA is negatively charged, so it moves towards a positive charge when you put it into the gel and you charge the setup. The DNA moves in a typical manner because this gel is actually not completely solid, right? It actually has sieve-like things in it. So DNA is able to actually penetrate and move forward when it gets the charge and the appropriate ionic environment, right? So depending on the time it's been put up, the DNA will migrate. The DNA with the smallest size will move the farthest away, and DNA with heavier will remain closer to where it's been added, right? Based on this, we can identify whether your amplification has occurred or not because you can visualize these bands. These bands are your target sequences. Remember in PCR, the target sequence amplified was much more in amount compared to the variable length, right? So variable length are not able to interfere at the end. It is the target gene which can be easily seen here. This is a DNA ladder. This is just added for reference, which will, since it's been added at the same time, it will move at the same speed, and we know what is the base pair size of each of these fragments. So if we can see the farthest, this is 100 base pair, this is 200, 300, 400. So in comparison, we can tell that this sample is actually roughly about 300 base pair. This is slightly more than that. And this one here is nearer to 200. This size was actually about 193, right? So this is how a comparison can be done. Remember, this is the easiest method available, but not the most specific one, right? So let's see a small video.
Welcome to this video guide, intended for use alongside the third
agarose gel electrophoresis as well. So for doing a gel, a scientist has taken a control sample, a loading dye. So what is a loading dye? A loading dye needs to be added to your DNA, which needs to be run on the gel, because it has various advantages. One is DNA itself is very light. So when you add into the gel, inside that well, it's lighter, so it might float away, right? So this dye, first is it adds weight to your DNA, the amplified DNA, so it settles to the bottom, and when charge is applied, it is able to easily migrate. Other thing is DNA cannot be visualized by naked eye. So how far your DNA molecule has moved into the gel, you cannot comment just by looking at it. So when you add the DNA, this dye to it, this dye will migrate with the charge itself, right? So you can see approximate where exactly your DNA has moved to, and it is not run away or run off your gel actually, right? And another thing is, you in the gel itself, you also need to add another dye, which is the fluorescent dye, right? How will you identify it? It is actually done by adding some fluorescent dye in it. Most commonly used is ethidium bromide, and this dye will actually bind to the DNA, and when you see it under a fluorescent meter, you can actually see this bright colored bands, right? So other than that, you also need buffers, which has ions in it, because the buffer or the ionic water is the ideal milieu for electricity to pass through it, right? So we can see here, this person is pipetting your amplified DNA as well as the loading dye and adding it into that small tube. Right, this loading dye generally have actually two dyes. One is leading strand, one is lagging dye, right? Leading and loading dye. There are different formulations. Some have bromophenol blue and some have other dyes in it. So we have, you can see at this small flask, we have measured the amount of agarose that we need to add inside this flask, and we add the buffer in it. Because first, this agarose is present in powdered form. So first we need to actually, to prepare the gel, we need to melt this agarose and make a liquid formulation here. So depending on the thickness that you required for your sieve, depending on what is your size of your target molecule, what is the base pair size, that much percentage of agarose needs to be prepared, right? So ideally we prepare 1 to 2% of agarose. So if it's a smaller molecule, you'll probably need it to be actually more thicker and vice versa. So we can see that she has added this into a microwave oven, and this is just to heat it actually simply. So just for a couple of minutes it's heated, and here we can see it's still a bit turbid, so it has actually not melted properly right now. We can see some particles here, so this step is now being repeated here. So now you can see it's quite hot. So now we can see a clear liquid here. So the agarose has completely melted, and your formulation for preparing gel is ready. So this molten agarose is now added into a gel tray. So this is a gel tray, nothing but something where you can prepare a rectangular block. And as it is still hot, you need to add it because it very rapidly cools down, and remember, it solidifies at room temperature. So this needs to be added very quickly. You can see the placement of the comb here. This needed to be done at this stage only, because after it's solidified you won't be able to do it, right? You'll break the gel if you'll try to make holes in it. So these wells will actually create deep grooves inside the gel. So she has added it and allowed it to cool down completely, and the gel is, after it you can see that it is clear, translucent in nature. After it is solidified, we are removing the cones and we can see the clear gel. So this is the setup which is ready. You have the electrodes here. And you can place the gel tray inside this apparatus. This is a gel loading apparatus. More buffer needs to be added. As we just said, ionic environment is needed for movement of electrical charge, and it will help the DNA to move through the gel easily. So now your amplified DNA, which has come out after the PCR with the loading dye, needs to be added. So she's pipetting it into the well. Simultaneously, we'll also add a DNA ladder. DNA ladder is nothing but already a reference ladder which has all the base pair which are already known, and we know their exact size. So we can see how it's added. The color given is actually due to the loading dye. Otherwise, the PCR itself was very clear. The PCR, the amplified DNA inside it. Right, with precision, it's needed to be added inside the well, passing through the liquid, which buffer that we have already added. So now the system needs to be closed and the electrodes needs to be added. Now this movement of DNA molecules needs to be set at a fixed voltage, electricity for a fixed time, right? Again, the choice of identifying the voltage is all dependent on your DNA molecule, right? We know about the size. The smaller molecule tend to move much faster. The bigger molecule, you just saw the small bubbles which are being formed. This is what happens when an electricity is started passing through the buffer. You can see these small bubbles coming. So when you move it, you can see these small strands, the larger fragments nearer to the well, and the smaller fragments at the end. On completion of electrophoresis, we turn off and we discard it carefully. Remember, it has the DNA binding dye also in it. So, and ethidium bromide has certain disadvantages that we'll just talk about. Right. So ethidium bromide, as we talked about, it's one of the intercalating dyes. Other examples are propydium iodide. You have other dyes like DAPI, which is a minor groove binders. It is just indicating the position where it binds to the DNA. It is non-specifically just binding to a nucleic acid. Intercalating binds between the linkages. The minor groove and major groove binding dye bind at the appropriate position. The problem with ethidium bromide is actually that it is carcinogenic. So theoretically, all DNA binding dyes have some potential, but actually most of the labs now try to discourage use of ethidium bromide, and we have now moved on to alternates like cybergreen and picogreen, which are actually much safer. for this purpose, right? There are different modifications of PCR techniques like reverse transcriptase PCR, which is being used, nested PCR, multiplex PCR, and real-time PCR. The reverse transcriptase PCR is actually the one which uses the PCR in 2 steps. It first converts RNA to DNA, and then this DNA is amplified by PCR method. There are various modifications of PCR which we are not going to cover in this lecture. Like once I tried to enlist, and there are more than 30 techniques, right? The most popular ones are actually these one, and we'll talk about those in brief. So as we just talked about, reverse transcriptase PCR is done for RNA, right? So it's actually the one which is most commonly used where we are talking about detection of RNA viruses like the SARS coronavirus 2 recently. So all of these assays are actually RT-PCR assays because it first converts RNA to DNA and then DNA is amplified. This can also be used for gene expression analysis where you're trying to actually detect the mRNA, right? This can be done in a single step, that is in a single tube, or it can be done in 2 steps. The RT-PCR separately followed by PCR, right? Another modification is the nested PCR where you actually do 2 PCRs. One is the external PCR and the internal PCR. In the first step, you take an outer set of primers, you identify the bigger region, and with this amplified product, you perform the second PCR. And here the primer is to your main target region, which is the internal one. Why this is being done? Because this method actually tend to have an increased sensitivity and specificity over the single assays, right? Multiplex PCR, as the name is suggesting, other are monoplexes where a single target is being used. Multiplex has multiple target in it, right? So this is diagnostically very useful because with the one amplified cycle you can actually detect many targets, right? For example, somebody is trying to do a respiratory virus panel and you think there could be 4 common respiratory viruses, could be your provisional differential, right? So you can actually do PCR for all of these together in one tube. There are several again advantages and disadvantages of this methodology because it compromises slightly on sensitivity, but its usefulness is much more, right? And while you are standardizing the assay, you need to be very careful that the primers do not actually self-bind or bind to each other, right? So the standardization is actually quite unique in this multiplex PCR. So for example, our lab used to do a multiplex PCR for dengue, chikungunya and Zika virus in a single tube, right? This nested multiplex format is currently are available in various commercially available cartridge based formats, right? Where you can add the sample, you can load it, and this goes through the cycle of extraction, the external PCR and the internal PCR in a single cartridge, right? This is a small machine. This cartridge can be fitted into this machine. This machine provides the milieu of the thermal cycling, right? And it has a detector which will also detect whether the amplification has occurred or not, right? This is the example that I'm giving you for BioFire or a film array. This is a cartridge based panels which are now available, right? So probably in the practical class today we'll just show you the pic and the demo of this cartridge as well so that you get the idea of the approximate size. Coming on to real-time PCR. So in conventional PCRs that we have talked about, it required a post-PCR analysis, but not so in real-time PCR. That is why this has become much more popular because in a post-PCR, the tubes need to be opened and manipulated. So anytime when you are doing it with a high amplified molecule, there is a chance of contamination, right? So, and also the time is also there, which is being wasted. So in a real-time PCR molecule, the target DNA amplification is occurring simultaneously by fluorescence based technology detection, right? And this is occurring in real time. This real time is as you see the cricket scores in real time, right? You are detecting something as it is happening. So this, what it does is, other than the primers, the forward and reverse, it actually uses another sequence which is called probe, which has a fluorescent dye attached to it, right? And this dye, when it attaches, will give this fluorescent signal which will be measured by the machine, right? The real-time PCR also has another advantage. Other than giving detection at the same time, it can also be used for quantitating your molecule, right? This DNA molecule can be quantitated into that into the template DNA that you have added. Based on it, you can actually give viral load or the load of your DNA or RNA in the original sample submitted, right? So you can actually quantitate the number of your organisms into the sample, right? This has huge application because it's been used in HIV, hepatitis B, hepatitis C, cytomegalovirus, BK viruses, and it has used in identifying development of resistance, for prognosticating, and for following treatment response, right? So how does it does that? You can actually use a set of known standards or calibrators in the real-time PCR together. This standards will actually create a curve, and they will create this is the sigmoid curve, which is what you get when you perform a real-time PCR. If you have used known standards and you chart it, you can actually get this linear line, right? And which has a certain mathematical formula, and this formula can then be used to identify the load of an unknown samples which has been added simultaneously. Remember, it has to be done in a single plate or tube together, right, in the same reaction mixture. So this is how we also quantitate in real-time PCR. When you get a real-time PCR result, there are certain things that you look for. You look for the baseline. This is the non-specific part that the machine is going to calculate fluorescence signal above it. And there is a term that we are using, threshold, based on which we measure CT or cycle threshold. This is again this term that due to COVID has become very popular. So CT is nothing but the number of cycle at which the fluorescence is being detected above the threshold value, right? So for example, if for this sample, so these are the cycle numbers, this is the fluorescence signal. So for example, if this is the sample, right, and this is how the machine has set, and you have actually manually adjusted, this has to be personally done by a person because the machine auto adjusts, and then a person has to manually adjust this threshold. So this is the line where this threshold, this is the cycle number at which the fluorescence of the sample overcomes the threshold value. So here we can see it is just above 20. So this would be roughly about 20. So we'll say that the CT is 20, right? So this is all about PCR and its modification, and right. So for further applications, I think we'll continue that in the second lecture, Molecular Biology Technique by Dr. Urvashi Singh. So thank you so much, and if you have any query, you can please send it through your CR, and continue giving your application though for the next lecture separately, right?